I'm going to introduce we have two TERI Talk speakers this morning. So the first one is Francisco de la Vega, who is the Chief Technology Officer at Galateo Bio. So Francisco, the stage is yours.
All right, good morning. Today, I wanted to talk to you about one of the topics of interest in my career, which is about how people intermingle and how people migrate, and what are the implications of that mosaic of genomes that we carry within ourselves and that we make by going around to places, and why this is important for why global diversity is important for precision medicine and equity. But first, I want to give you my PSB story here. This was my first PSB paper in 1996. That was the first time the conference had a proceedings. I guess it's not officially the first year of PSB, but it's canon, the first year that we have proceedings. In the proceedings online, 1996 is the first one. There was a book, but not online. So this is the second year. I came here from Mexico when I was assistant professor, and I convinced my boss to send me to Hawaii. And thank you also for some support from PSB to make that possible. That pushed me to try to contribute to PSB over the years as much as my bosses let me. So anybody of you in industry, please contribute to PSB, because there's people that want to come here, and that's the way to make it happen. In those days, I was talking about education in the internet, how we could teach computational biology. That was a new discipline then. If you're interested in doing some archaeology and see what are the weird things that we did at that time to teach computational biology, you're invited to look at that paper. But coming to PSB is addictive. So here's the other sessions that I came, and I chair 18 in total, so I'm at TERI. And I had the collection of books, I think, until they stopped printing them. But after that, I immigrated to the United States, to industry. And I worked in several companies over the years, for a long time in applied biosystems, and more recently in small startups. And I developed a lot of technology there, but today I'm not going to be talking about specifically of those technologies, but about how during my career in industry I was able to pursue a topic of interest and always been able to have a feet on industry and a feet in academia. And the topic that for somebody like me that was important is how population demography impacts human genetic variation, and what that tells us about the impact of this variation in disease incidence, outcomes, and with respect to therapies. So one of the first things that I did at Applied Biosystems was to collaborate with others, including Debbie Reich, to develop a microsatellite map to take advantage of the mixture of recent mixtures between populations to map disease genes. This is called admixture mapping. And so we selected a map that you can see in the figure here, the calculated power to find disease genes when there's more prevalence in one population versus the other. And the maximum power you can see here for African descent populations in the Duffy locus, which is fixed in African populations. A number of discoveries we've made with this map, including a locus for prostate cancer in African American individuals, and so on. But a problem of these technologies was the resolution. We really were able to just find big chunks and figure it out what's going on there. It took a lot of work. So just after human genome was finished, the first thing that the community said, well, it's great to have a reference, but there's genetic variation. What if we go about and map the genetic variation? So several efforts ensue after that. One effort that I was involved was a private effort. At Applied Biosystems and Celera, we decided to do the first exome genome project with 40 samples. Japanese, I think it was Japanese and European initially, descent. but it was done brute force with Sanger sequencing, with one amplicon for every exon. This little project cost about $100 million. On my side in Applied Biosystems, we developed TACMAN assays for SNP genotyping, tools for research. We validated 200,000, and along the way I thought, maybe we can explore what's the linkage disequilibrium differences between the populations in our panel. And so we had a panel of African Americans, European descent, Chinese, and Japanese. And here's a picture of the paper that we published in 2005 of 3 chromosomes. This is chromosome 22. And what you see here is the chromosome physical distance on the X, but the distance between markers in linkage disequilibrium units. And what you can see immediately is that there's a lot of shear slopes and valleys, which means the shear recombination sites across populations. But you can see that the African American individuals have a longer map, which clearly talks about the depth of the population from Africa. So this was an interesting finding, but the larger research community started to organize to take advantage of the new next generation sequencing technologies and have a map of genetic variation at the base pair level that was built on the HapMap project. And initially, that was, we call it the 1,000 genome project, was to sequence 1,000 genomes. It was very expensive then, and 4 populations were selected basically from the HapMap. An African population from Yoruba, Chinese from Beijing, European from Seth, and Japanese from Tokyo. And the question was, is this sufficient? Is this really a representation of the genetic variation across the globe? Clearly it's not. So in collaboration with my partner in crime here, Carlos Bustamante, I was part of the steering committee at the time of the 1,000 Genome Project as the company contribute to this effort initially. Esteban Bochar as well. We look at the data on the current association studies and we said, hey, there's not a lot of diversity here. And this is a major problem when we are gonna try to translate this into precision medicine. So we wrote this commentary in Nature, and we showcased that from all these studies so far, only 4% of the people recruited to these studies were non-European. So clearly, there was something to do about that. Meanwhile, in the 1,000 Genome Project, there was a debate. Well, if we expand this, who's gonna pay for it, right? And Carlos Bustamante, myself, and others were pushing to improve that. But I told to Carlos, hey, what if we just sequence some admixed genomes to showcase what we can learn from these diverse populations that so far has been avoided in genetic research. No, it's too complicated. So we sequenced the first Mexican, American, and African American genomes, demonstrating how these could be of interest. Ultimately, we published this in a paper in 2012 with 12 additional samples. And we think this effort contributed to the decision, the 1,000 Genome, to add more populations. But we learned a couple of interesting things there. Typically, we see these PCA plots where each of these clusters are populations. But now, something we wanted to do that is to look more inside of our own genomes. And we can see this mosaic here to the right, because at that time we implemented the first local ancestry methodology to be able to distinguish which chromosomes come from ancestral population in individuals of recent admixture. So you can see Puerto Ricans on the top, we had just a few trios, Mexicans, Americans from LA in the middle, and African Americans from Southwest on the bottom, so you can see the colors represent some of the ancestral populations. And we also were able to see that within each of our genomes, we also have the history of the entire human migration diversity. This method developed by Richard Durbin, pairwise sequentially Markovian coalescent, was applied to one sample. When he published this in Nature, we applied it to our samples that we had access to. And we can see as we go far on the x-axis, that's generations in the past. We can see a population bottleneck that is very severe for out-of-Africa populations, which is what we kind of already suspected, right, that shapes genetic variation. But now with local ancestry, we can split apart those genomes and do the same for the ancestral population. And so here we got the first view of the population bottleneck of Native American ancestry in those Mexican genomes, which is a first thing that we were able to do. The other thing is our genomes also tell us about when those admixture events happen. So to make a long story short, the distribution of the length of these admixture tracts tells you about when and whether many pulses contribute to the admixture. So we recapitulate here the history of colonialism in the Americas and the forced migrations for African Americans that we know led to the current admixture that we have today. So the 1,000 Genome Project became the 2,504 Genome Project at the time. We were able to sample a lot more populations, still not a full spectrum, but we added a lot, and this is a great resource. Even today, this is the only resource of genomes that you can download. A high schooler can download with no paperwork, having to go to IRB and nothing, right? Everybody uses for developing new technology, validating that, so it is a great resource. We clearly learned and discovered a lot more genetic variation, and clearly those populations on the right, which come from Africa, have the highest level of genetic variation, and clearly that tells us that there's a lot of things to discover in these populations. On the right-hand side, again, this method to look at population bottlenecks. Now we have a much bigger picture of population expansions and contractions into the past with all of these genomes. But the question remains, have we done sufficiently since then? And it turns out that since our commentary in 2011, I just checked this a few days back, in 2014, we just increased from 4% non-Europeans to 9% non-Europeans in the recruitment of people that go into genetic studies, which is telling us that we're not yet there. We're not yet there. So there is an unmet need to continue to increase this, because something I'm not showing here, but if you see at the proportion of discoveries, they actually come disproportionately more from other populations, even if the recruitment is like this. Then I'm going to tell you about a few other things that I did more recently. I spent 4 years in Tempus, and a company focusing on oncology, and one of the things that I wanted to look at there, given the huge database of multimodal data, is the topic of cancer disparities. It is very well documented. There are disparities in the incidence and outcomes in particular that are based on race and ethnicity. And clearly, those disparities are mainly derived from socioeconomic, environmental, structural factors. But there's some evidence that there is some of these disparities that could be at least mediated by biological changes. And we thought that better understanding of that could help us to figure out what's going on and what can be done about that. So we set to analyze 100,000 cancer patients from the Tempus library, which we have somatic and germline data. And we selected ancestry informative markers, and we inferred the global ancestry on these samples. So you can see here they are separated by cancer type, and the colors are the ancestral reference populations to which we compare these ancestry informative markers. You can see in dark blue is the European descent. So most of our patients in Tempus are clearly European descent, but because of the scale of the operation, now we can have enough patients, actually unprecedented number of patients that otherwise would be hard to have of cancers from more diverse populations. And we also find now complex levels of admixture, some topic that we explored last year, one of the presentations on how these admixture levels are changing over the more recent generations. So we were able to study the association within the rates of somatic mutation in cancer genes and ancestry. And this is just the significant results for some set of cancers that we have larger sample size. And we were able to find what we already knew. There was in the literature already descriptions that, for example, mutations in EGFR are more frequent in East Asian individuals, that mutations in APC, that was in lung cancer, that APC in colon cancer are more frequent in African individuals, but for example, mutations in BRAF are less common in African individuals. And that has consequences in targeted therapy because some of the targeted therapies are targeting these kind of mutations. But as you can see, we were able to find a lot more because it was an unexplored question. Some of these could be really interesting and useful. For example, we find association of somatic mutations in the beta-catein gene, CTNVV1, that turns out they are a candidate biomarker for the effectiveness of new anti-cancer drugs. So, and by the way, this is in lung cancer, and people said, no, there's no mutations in this gene in lung cancer. Sure, in Europeans, there's no mutations, but you look in other populations, and what is rare in one population may be common in another population. So that's what we're trying to find, something that is rare in one population, but is common in another, and that can help everybody else, not only that population. Now, the question always remains, is this just social determinants of health? This is some data from, not from me, from researchers at Dana-Farber. Giancarlo Jan presented this research a couple of years where in lung cancer, she found a correlation between Native American ancestry, that was in a Hispanic cohort, and mutations in key cancer genes. I'm just showing here EGFR. You see the more Native American ancestry, and this is statistically significant, the more EGFR mutations. And the question again was, is this just associated but not causal? So something that I think was great that they did is that they used local ancestry again to try to see if the signal was focused in some regions of the genome, and they indeed found that. If it was just social determinant of health, everything will be lighting the same way, but now there's some candidate loci that we could investigate. Finally, in the last 4 minutes, I will tell you about an effort that we're doing in Galatea Bio, the company that I joined in the summer that was started by Carlos Bustamante, where we're trying to take advantage of genetic diversity around the world for biopharma, to empower biopharma research and medicines that could work for all of us and translate this into precision medicine. So the CAPA Consortium is a consortium on asthma among African American populations in the Americas, started in 2011. I was also part of that collaboration with Carlos when he was at Stanford. So the aim was to identify loci for asthma in this population of African ancestry, in particular those of the diaspora in the Americas. And they were able to find new genes, confirm genes that were also in both populations, European and African American, and discover new loci. But they kind of, to close this project, at Galatea, we decided to collaborate with them and sequence 500 of these samples with long-read technology. Some of these samples were already sequenced with short-read, and we thought, hey, how about structural variations? Those are important, right? But we cannot discover them very well with short-read. So we are in the process of sequencing a variety of samples representative of this collection, in particular populations that have been really underexplored, like, for example, the Garifuna from Honduras, which has African ancestry. In fact, also in Honduras, there's another population called the Mosquito, which has East Asian ancestry. So they are very interesting populations. And in this PCA, you can see the black squares here. Those are our populations, and they actually represent non-genetic diversity, but novel genetic diversity. So we are very excited about what we can find. Here's just a glimpse on the structural variation discovery that we're doing in these samples. We see the patterns that we're supposed to expect. Longer events are less frequent. You can see the peak for allo repeats online. If we look at the numbers, we can see that populations that have higher African ancestry have higher diversity in structural variation as well. Most of the diversity in the 1000 Genome Project was described were SNPs. We also did some structural variation, but not great. So now we have a wonderful resource which we are pursuing a series of tracks. Obviously, catalog structural variation, see which of these overlap with disease genes, select some of these samples for T2T assemble to contribute to the Pangenome project. Pharmacogenetics, what new alleles are there? We just develop the drugs in the US or in some selected countries, and we think it's going to work well for everybody in the world, but is it? And finally, HLA alleles. Clearly, there's a lot of interaction with the infections and other diseases, so we think we're going to find new haplotypes that could be of interest into how these populations have been evolving over time and the environmental interactions with them. So with this, I want to thank people that have helped me along the way. Carlos Bustamante, which I know for 25 years or more, who has been a important part of all of this. Andrew Clark, which also was collaborator with Carlos. And in Applied Biosystem, Dennis Gilbert had our Isaac. Ken Kidd, a great collaborator, and Michael Honkapiller, which allows us to do this at Applied Biosystems. At Tempus, my team, Brooke Reed and Janet Poulot, help us in all this. We have one paper published. We have 2 more on the works. At Galatea, my new friends there and my team in the Kappa project, Paige, Mike, Babak, Pavel, and also Catherine Barnes, which is a PI in the Kappa project, and obviously the Kappa consortium members. Some of the art here from Lynn Feldman, great artist, if you want to look at that. And if you see me on the road, say hello. Thank you.